Thank you so much. I would like to thank the EHF for this opportunity to come to Israel for the first time and enjoy the beautiful country and the great food and the beautiful weather. I'm glad some of you were able to come back after lunch because I'm sure it was very tempting just to continue to stay out and sit in the sun. But I also am glad that you're here so I can tell you a little bit about what we do know about environmental endocrine disrupting chemicals, or EDCs, as I'll be calling them throughout the course of my talk. So unlike the conversations that we had during the first half of the day, my patients look a little bit different. We have a rat model for endocrine disruptors in the lab that we've been using now for probably almost about 15 years. Excuse me. And the rat model turns out to be a great model for understanding effects of EDCs on hormonal systems and on neurobiological systems, because the molecules that control reproduction across the life cycle that are both in the brain as well as in our gonads and the rest of our body are virtually identical between the species. And so how the brain and how the gonads interact to control reproduction is very highly conserved. In fact, it's really virtually identical in all mammals. Excuse me, I'm just going to grab some water. Okay, so to continue the rather pessimistic news that we started this morning, I'm sure you all realize that we've contaminated our world. And although there are some people out there, and I'm generally a very optimistic person, but there are people out there who think, well, we can clean it up and we can make everything go back to the way it was before. But I think that's obviously very naive. And although we can do local remediation, it just simply is not possible to go back to the way things were. And while clearly there are areas of the world that may be more contaminated than others, and other parts of the world that have been more successful in cleaning it up, the reason I showed this slide is that it highlights the point that people who live in parts of the world, including in the Arctic and the Antarctic, where there's not heavy industry and use of pesticides and lots of industry and industrial chemicals, these people are still being exposed to environmental EDCs. We find EDCs in the bodies, in blood, in fat of humans, in sea mammals, marine fish, and so on and so forth, due to transport of these compounds around the world, both through migration of the animals as well as through the air and water currents. So the EDC field and air pollution fields actually overlap a great deal, because we know that EDCs can be transported around the world through the air. So let me give you a working definition of EDCs. Recently, I worked with a group of colleagues from around the world, and we wrote a paper about sort of the recent issues that are important about endocrine disruptors and their effects on human health. And in the course of those conversations, we realized that we really needed to redefine what an EDC is. Previously, people in the United States were depending upon a rather old definition by the US EPA of what an endocrine disruptor is, and it was rather narrow and it had a number of conditions. And we decided to redefine an EDC, as shown here, as an exogenous chemical or some mixture of chemicals that can interfere with any aspect of hormone action. So I've provided a short list of what are some of the common EDCs that are out there today. Of course, there are industrial contaminants. In my lab, we primarily study polychlorinated biphenyls, or PCBs, pesticides, pharmaceuticals when they get into our environment and are obviously being used for purposes other than for which they are intended, can become EDCs. They're in fish. People drink water contaminated with pharmaceuticals. Plastics and plasticizers, including BPA and phthalates. We could even consider some phytoestrogens to be EDCs, especially if they're consumed in very large amounts through supplements. And heavy metals can also act as EDCs through actions on hormone receptors. And we have many examples showing that both humans and wildlife are being contaminated by EDCs. And I don't have time to tell you all the stories, although there are obviously a lot of very interesting stories about how EDCs were discovered and initially how they were described as having effects on reproductive systems, causing infertility, causing abnormalities in structure, especially of reproductive systems, for virtually every member of all of the vertebrate classes. We also know in humans that exposures to EDCs can have adverse health effects. And a lot of that evidence has come from high-dose exposures where there were either accidental explosions or food contamination or even pharmaceutical exposures during pregnancy where women were exposing their fetuses to high levels of hormones, unintentionally exposing the fetuses. And we know from these examples of, for example, PCBs contaminating cooking oil, explosion of a factory in Italy about 25 years ago now that exposed thousands of people to dioxins, and diethylstilbestrol, which was an estrogen pharmaceutical given to women in an effort to avert miscarriage, which, as it turns out, DES actually does not protect against miscarriage. And it's estimated that millions of women exposed their developing fetuses to DES, which is a highly estrogenic compound, during their pregnancies. And in those days, 50, 60 years ago, nobody thought that any of these compounds could get to the fetus because it was believed that the placenta could protect the fetus. But we now know, and DES was one of actually the first cases to prove that the fetus is not protected and that hormone or hormone-acting compounds can sometimes very easily cross the placenta and get to the developing fetus. Okay, there we go. So there's also now really good epidemiological evidence in the US based largely on a national data bank that's called abbreviated NHANES, where the Centers for Disease Control, or the CDC in the United States, has measured almost 300 chemicals in banked samples of humans whose health and who had been extensively surveyed and had been examined and gave these tissue samples. All of these compounds were measured, and just as an example given to me by Jerry Heindel from the National Institutes of Health, up to almost 300 chemicals have been found in cord blood, in breast milk, and when people, adults, are tested by the CDC, basically 100% of people have these industrial chemicals in their bodies. So what I'd like to do now is turn to some of the research that we're doing in my lab, and I guess you can consider it to be an example of the type of basic research that's going on to determine the health effects of EDCs. Now, obviously, in the end, we want to understand what are the effects of EDCs in human beings and what are the potential adverse outcomes for ourselves, for our children, our grandchildren, and so on. But on the other hand, we can't do experiments on humans, which is a good thing. So what basic scientists have been doing is turning to these animal models. As I told you, we work with rats in the lab, and we look for systems that are highly conserved amongst mammals so that we can infer from the animal data to what the probable health effects are in humans. Now, my research interest is how does the brain control reproduction or reproductive hormones, and in turn, what are the actions of these peripheral reproductive hormones, such as estrogens, progesterone, testosterone, and so on? These hormones circulate through our body, and clearly they're very important for reproductive processes, but these hormones can also get into our nervous system and provide feedback actions onto the nervous system. So my basic science interest is what is called reproductive neuroendocrinology, namely the link between the nervous system and the endocrine system in the control of reproduction. So that is basically summarized pictorially here, where we're looking at a cross-section of the brain, and I've highlighted in blue the region of the brain that's known to be most important for hormone and endocrine regulation, which is called the hypothalamus. All animals, beginning with fish, all the way through all mammals, have a hypothalamus, and the hypothalamus controls reproduction in every vertebrate. So normally the hypothalamus will function across development to drive the pituitary and ultimately the gonads to regulate development. And what I'm interested in is how EDCs that may act like hormones or may perturb hormones may influence these processes. And then I look at these endpoints in my animals, such as the hormones of the animals, reproductive functions, and finally the behaviors of these animals to determine the outcomes. So here's my model. We begin with exposures to environmentally relevant levels of EDCs. We take pregnant rats during gestation, expose them to low levels during a short period of development. The levels that we're exposing the animals to are estimated to be similar to what is out being, or what wildlife are being exposed to and what humans are being exposed to as well. And we've chosen a short period of embryonic development when the brain is developing and when the brain is particularly responsive to gonadal hormones normally during development, and when we predicted that disruptions of this process would be most likely to perturb this process. So I want you to consider that there's this very short critical window of development during which we have this exposure period, and we are not causing toxicity, we're not killing the fetuses, we're not causing them to develop 3 heads or to do anything that really is abnormal, but what we're doing is exposing them to these low levels and then following them across the life cycle. Once the babies are born, they go through puberty, they become adults. In females, they have reproductive cycles, ultimately into aging, and determine what are the potential outcomes and what are the mechanisms which we can look at in our animals that were being invoked by these exposures. So don't worry about this picture, but I want to give you a framework for how the brain controls reproduction. So imagine now that you're looking at the brain, focusing in on the hypothalamus and focusing further in on subsets of nerve cells in the hypothalamus, neurons, that we know are responsible for the control of reproduction. These neurons produce a neuropeptide or a neurotransmitter that's called gonadotropin-releasing hormone, or GnRH. And this GnRH peptide is the single neurotransmitter that goes from the brain to the pituitary gland, which then subsequently stimulates the ovary and the testis. So there is a single molecule from our brain. It's a neurotransmitter, but it acts like a hormone because it travels down to the pituitary gland. And it is identical between humans, rats, dogs, cats. Every mammal has this exact same GnRH. We know that changes to the GnRH neurons in the hypothalamus as we develop are critically responsible. They are both necessary and sufficient for driving the onset of reproductive development in puberty, for controlling reproductive function in adulthood, and for maintaining fertility until we become very aged. We also know that the GnRH system, being part of the brain, of course, no cell in the brain is acting in isolation. So the GnRH neurons are receiving these inputs from other neural cells, other glial cells. There are synapses amongst these neurons. And these neural cells that make these inputs to the GnRH neurons provide information to the GnRH cells about the environment. So what is the environment? The environment is everything else. The environment are all the hormones in our bodies. There are physiological status. They're the external environment, including chemicals, including air pollutants, including your social environment, your family. All of that environmental information gets coordinated through these converging neural inputs that come from other brain regions that ultimately target the GnRH neurons. There was only about 1,000 of them. Everybody in this room has about 1,000 GnRH neurons. Those 1,000 neurons are what are driving our reproductive function. And what I'm interested in is how does this network develop, and what are the consequences of environmental exposures to EDCs during a very small exposure window prior to birth when this network develops. This slide also serves for me to remind you that the GnRH neurons then act through the pituitary gonadotropins and then ultimately onto the gonads. And the gonads themselves are then producing steroid hormones that circulate through the body, but also feed back to this neural network and thereby communicate back to the brain information about the organism's hormonal status. So we have a somewhat complicated hypothesis, which I'll just share with you quickly, which is that we believe that EDCs perturb hypothalamic development through what we call reprogramming of the development of this neural network that controls reproduction. So I'm going to take you through a series of experiments that we've done to demonstrate how the EDCs are having these effects and what we know about them. So to begin with, just a quick introduction to our experimental EDCs. We selected a mixture of PCBs, or polychlorinated biphenyls, which are called Aerochlor 1221. And we actually got interested in PCBs. I began this work when I was on the faculty at Mount Sinai School of Medicine in New York City. And as some of you may know, the Hudson River, which flows right by Manhattan in New York City, is highly contaminated with PCBs due to heavy industry upstream in upstate New York, on the upper part of the Hudson River. And so I always assumed, being naive, that nobody would ever touch the fish in the Hudson River because everybody knew that it was contaminated with extremely high levels of PCBs. But actually, quite a lot of people fish in the Hudson River, and people in New York City are exposed to quite high levels of PCBs. And then we're also exposed to PCBs through other contaminated food and water sources. Fish tend to have the highest levels of PCBs. So although the PCBs were banned in the late 1970s in the US, everybody in the US, and I'm guessing everybody probably in this room also, has some body burden of PCBs. They're persistent, they bioaccumulate, they get into the food chain. We know that adults consume PCBs. That's probably the major mode of exposure. And what I'm concerned about is transfer of the PCBs to the developing fetus when the mother has been exposed in some way, looking at the neurological and the reproductive consequences. So here's my model, and it's so nice to be able to work with rats for the reason that we can control everything. We can start with the pregnant rat. We expose her late in gestation, and in fact, I'll show you on day 16 and 18 of gestation, which is the beginning of the third trimester, which is referred to as a critical period of brain sexual differentiation, because during this period, there are big sex differences between the males and females in how and the levels and the ratios of the hormones that our bodies and our brains are exposed to, both in rats and in humans. And so we chose that period for the exposure to one of three. In the beginning, I'll talk about three treatments. One is a vehicle. Of course, you have a control vehicle treatment. Some of the animals are treated with the PCBs, the Aerochlor 1221. And because we know that PCBs, or at least this class of PCBs, can act at least in part through actions on estrogen receptors or in estrogen-sensitive pathways, we also treated a subset of animals as another control with estradiol, just exogenous estradiol, as an estrogenic control. So this happens during the the beginning of the third trimester. Babies are born, they look completely normal. We don't have any birth deficits that we can see. And we follow the animals through postnatal development. And I've highlighted this period here because the first experiment I'll tell you about looked at the animals beginning about 15 days through about 90 days after birth, selected because puberty happens between about day 30 and day 45. So this is the sort of the childhood period for the rats. This is early puberty and adolescence. They go through puberty and they attain adult reproductive function at about 45 days. So they also develop very quickly. It's a great model to work in the lab, right? Because our animals become adults by the time they're 2 months old. And we also compare them to animals that are full adults at about 90 days of age. So just a couple of points of interest. We know in the United States, and I'd be interested to know what the data are in Israel, but in the United States and in Europe, puberty in girls is happening a little bit earlier over about the last 30 or 40 years. And we looked at the timing of puberty in our animals. So just to orient you to my slides, because they're going to be organized sort of similarly for the next few slides, I'm going to show you data for females on the left and males on the right, keeping in mind these animals were prenatally exposed. There were three groups, the vehicle group, the estradiol group, and the PCB group. And in this case, we looked at the age of puberty, as we can monitor puberty by secondary sex characteristics in the rats. And it's pretty hard to change the timing of puberty in rats. So changing it just by a day or two actually can be highly significant as far as development is concerned, because their development is so accelerated. And what we found was that in the females, the vehicle animals go through puberty at about 35 days, and the EDC animals, both the estradiol and the PCB animals, go through puberty a couple days early. In the males, we actually saw a slight delay of puberty. And what we think is the explanation for these data is that the estrogenic EDCs, we know estrogens stimulate puberty in girls. So that's consistent with this finding. Whereas in boys, estrogens don't stimulate puberty. They can actually slightly retard the timing of puberty, and that's what we see in our males. So these rat data are actually quite consistent to what's going on with humans, and we do believe with humans that although there are many reasons why girls go through puberty earlier, we think EDCs are a part of that. I'd also like to talk about a little bit of the neurobiological mechanistic data, and I'm just going to summarize those data because I looked at 48 genes in the brain, I looked at a lot of proteins in the brain to see how those were affected by the prenatal EDCs. I'd like to acknowledge Dina Walker, who's the graduate student who did an enormous amount of work and did a great job. These are the gene names. I don't expect you to know what these gene names are, but these are the genes that were affected most profoundly by the prenatal EDCs in our developing animals. And I'll go tell you what they are in just a second, but what we noted was that the females were much more susceptible than the males to the developmental EDCs. And overall, the patterns of gene expressions that we saw in the females made their brains actually look more like the male brains in the expression of some of these genes. So just to tell you what some of them are, GnRH, I already told you about, is this neuropeptide that controls puberty. These are genes, kisspeptin and prodynorphin, are other neuropeptides we know are critical for GnRH regulation. Those were changed. AR is the androgen receptor. ESR1 and ESR2 are the two estrogen receptors. And these are additional estrogen and progesterone receptors. So there were many critical genes in the hypothalamus of our female rats that were changed by the EDC exposures in a manner that made them appear to be more like males. We also looked at protein expression. I'm just going to talk about 4 proteins. I'll have to go through these data rather quickly, but what we did in this particular study was look in parts of the hypothalamus at the estrogen receptors, kisspeptin, one of the peptides we know is important, and activated GnRH neurons, and how those were affected by the EDCs, done by Sarah Dickerson, another graduate student. And I'll just show you 1 or 2 examples. So this slide is organized just the same as the puberty slide, with the females on the left, this shows estrogen receptor cell numbers, and in control females, estrogen receptor expression is very high compared to control males, which is well known in the literature. Actually, this is a section through the brain, and each of these little black dots is an estrogen receptor. And this is a female and this is a male, and I think you can probably see females have about twice as much. When we treat the females prenatally with the EDCs, their estrogen receptor cell number is just about cut in half. And if you compare them to the males, and in the males there wasn't an effect of the EDCs, I think you can see the females are what we call masculinized. Their estrogen receptor expression in the brain is now more similar to that of the males. I'll flip through some of the other data because of time, but we also saw significant effects on another estrogen receptor, which is called ER beta, which was decreased in the females treated with the EDCs prenatally. And this was looking at the animals in adulthood. We also looked at kisspeptin expression, this important neuropeptide in females, much higher in females than in males. And females treated with EDCs had a big decrease in kisspeptin, not quite as low as the males, but certainly in that same direction. And finally, we looked at activated GnRH neurons as an indicator of GnRH function. We just did it in the females because for technical reasons, you can't do these experiments in males. And what we found was that in females, about 60% of the GnRH neurons are activated in controls, and only about 10 to 15% of their neurons are activated in the EDC-treated females. So just to summarize what I've told you, we know that developmental exposure, prenatal exposure to EDCs cause large changes in the developing hypothalamus. I've emphasized that these changes are primarily happening in the females during a period of sexual differentiation where we believe that their brain is becoming slightly more masculinized in terms of the expression of some of these molecular markers of the control of reproduction. And overall, the consequence of this EDC exposure is a disorganization of this hypothalamic neural network that controls reproduction. So what does this mean, right? So the females are not becoming males. They're not everything. Of course, I showed you the data that, as a neurobiologist, I find very interesting. But of course, there are many other aspects of this neural network that were not affected. We also saw some changes in the male brain that I didn't have time to talk about. Overall, we haven't turned the females into males. So what does it mean when we have this change to this neural network in the brain caused by EDCs? And in order to look at that, we just did a couple of simple experiments. We looked at reproductive cycles in the females when they were adults, and we also looked at their reproductive behaviors. Again, something nice about working with rats, you can look at their behaviors. So we know with rats, adult female rats normally have regular reproductive cycles, just like women. We have menstrual cycles of about 28 days. Rats have reproductive cycles of about 4 to 5 days. And so a regular reproductive cycle involves transitions from one day to the next. Each of these dots is representing a different reproductive cycle status with regular changes from day to day. And what we did with our animals, once they had gone through puberty, was ascertain what percentage of the rats had irregular cycles, where either there were protracted days without change or changes going in directions that you were not expecting for them to go. And our control animals, none of our animals had irregular cycles. There were 27 animals in this group, whereas in the two EDC groups, about a third to a half of those animals had irregular cycles. And we know that those neurobiological markers I was talking about are very important for reproductive cyclicity. So this is very consistent with the molecular data. The other thing that we were able to do with our females was put them through a mating test. So we did something called paced mating, which is a little bit different. So let me just take a moment to tell you what the test is. In a paced mating test, since our lab is primarily interested in female reproduction, it really allows you to focus on the female behaviors. Now, there's been really great physiological and behavioral work done by other labs showing that if you allow a female to choose the pace of the mating, to determine the timing, whether or not she's going to mate, and really to make the decisions about mating, that she actually is more likely to have reproductive success, as evidenced by pregnancy and having offspring. So this is very well established in behavioral neuroscience. There's a whole, it's a really interesting field. I wish I could tell you the whole story about paced mating. But basically, what we did with this test, and it's also a great test, and you can do it all in your own labs, because all you need is an aquarium, like a fish tank that's about this big, and you partition it in the middle with a piece of plexiglass, and at the bottom of the plexiglass are these holes that are cut out. And on one side we place a female, and on the other side we place the male. And the holes are small enough that the female can pass between the two sides of the chamber, but the male is too big to pass between them. So he can only stay here, but she can go back and forth. And I'm going to show you, hopefully my video will work. I'm going to try to show you a video showing how, and it's going to focus on the male side because it's not, when this female's by herself, it doesn't really matter what she's doing. But I'll show you the whole mating, the dirty movie of rats, I guess, to wake you all up after lunch. And what you'll also see as the female engages in mating, that every time the male mounts her or has an intromission or ejaculates, she'll engage in a behavior called lordosis, which is a receptive behavior where she'll arch her back, and you'll see that. The other thing you'll see is that after the male does something, the female always leaves that chamber and goes to her home chamber. So that is how she's pacing the mating. She determines how long she's going to spend on that side and how much time she's going to spend away from him and whether or not to mate. So let's hope this works. Oops. I'm not very good with a PC, so. What you're going to see if this works is the male side of the chamber, and there's a barricade, and then the barricade will get lifted up, and there you go. So there's the male, there's the female, barricade just went up. So you'll see the mount, the intromission, and the ejaculation by the male. This is the lordosis, where she arches her back and she's trying to get away from him. With the intromission, I also want you to notice that after he intromits, she'll run away right away. There she goes. And then at the end, after he does that about 10, 12 times, and then he finally ejaculates, and then she runs away. And he kind of collapses and takes a break. Okay, so we quantified a lot of behaviors, and I won't have time to show you all the data, but we have published this paper a few years ago now. And I'll show you just a couple of things that we noticed about the females that was really interesting. And it has to do with pacing and timing. The amount of time it took the female to return to the male after he either mounted or intromitted or ejaculated, and I'm showing you here ejaculation return latency, is significantly higher in the PCB group than in the control group. So it's about 30–40% longer for her to come back. This is time in seconds. So it normally takes her about 2 minutes to come back. Here it takes about 3 minutes. The other thing I thought was very interesting is that the number of trials it took the female to successfully mate with the male was also significantly higher, meaning that the females don't always mate in the first trial, and with the EDCs, it takes them even longer. So considering this is a controlled laboratory environment where everything is provided to our animals, they have a wonderful life, their reproductive success is changing in the animals that have been given the EDCs. And when you try to translate this to a naturalistic circumstance like being in the wild, where not everything is so perfect, where you have to be able to mate as quickly and successfully as possible, this actually could have a significant adverse outcome on reproductive success. So in summary of this section, we showed that reproductive cycles are disrupted, that paced mating behavior is disrupted, and we predict that reproductive success under other circumstances would be disrupted by the prenatal exposures. Now let me end by taking this to a little bit farther along the life spectrum, and I won't tell you the whole story, but I'll go through this quickly. The other part of my research interest is understanding the neurobiological control of reproductive aging. How does the brain change? How does the hypothalamus change as we age? What does this have to do with menopause in women, and presumably in men? What does this have to do with the loss of reproductive function with aging? And so in collaboration with a colleague at Rutgers University, Mehmet Uzumcu, I did a study looking at effects of early life exposures. In this case, it was to a pesticide called methoxychlor, and how that affected reproductive aging and the hypothalamic control of reproduction. What we found with his animals was that his rats went through very early reproductive senescence. They aged much earlier than control animals when they had been exposed to the EDCs. And just for the sake of time, I'll summarize what happens in the hypothalamus of these animals. We found a lot of molecular changes in the hypothalamus of these animals, and I'm not going to go through them, but it includes estrogen and androgen receptors, other neurotransmitter receptors, kisspeptin, and so on. Think about the fact that we treated these animals shortly before the time of birth, and these are the brains of animals that were like my age. So, at, say, perimenopausal, early postmenopausal age, they still had these brain molecular changes from that early life exposure. And that's really the bottom line of this part of the talk. So I'll show you, if I can get the slide to change, just two examples of two genes that were perturbed. One was the estrogen receptor in the hypothalamus. The other was kisspeptin in the hypothalamus. And those were actually similar to some of the genes that had been affected by the PCBs in some of my other work looking at young adults. And again, for lack of time, I can't really get into the mechanisms, but we also discovered that there are some epigenetic molecular changes. If you're familiar with this terminology, maybe we can talk about this a little bit later. But there are epigenetic molecular changes that we also find in the brains of these animals. So let me summarize. I've shown you that prenatal EDCs perturb hypothalamic development and this network of genes and neurons that controls reproduction. There are functional consequences. There are lifelong effects into aging. And although I didn't have time to talk about it today, we also know there are transgenerational epigenetic effects, meaning effects passed from generation to generation to generation. Okay, so I'll end by saying that I think there is some hope, since this is all so depressing, that by understanding these molecular mechanisms and by doing the basic science, we can communicate to the clinicians and the public and the media and the scientific societies and the organizations and tell them, here are the problems. There's really good science out there. I'm not saying mine is the only science out there. There's a lot of really good science out there that is supporting all of these conclusions. We also need to partner with green chemists and with people who have expertise in making the next generation of chemicals to ensure that we're not continuing to contribute to the problem. So I'll stop here, leave up the acknowledgement slide for the money and the people who have done all the work, and maybe there's a minute for questions. Thank you.
Thank you very much. It was a very interesting lecture. We have time for one short question. Please.
Oh.
So just speak loud.
If you speak loud, I'll repeat the question.
Okay. I was just wondering, during the first half of the day, you talked about standards. Now, do you see a threshold effect, or is it effective in any concentration?
So the question was, are there thresholds? So the answer is, there are no thresholds for endocrine disruptors. And that is possibly something we can argue about later. But when you consider that there are periods of life during development when our bodies are normally not exposed to any levels at all of some hormones, or we have receptors for these hormones, yet our bodies, or say in pregnancy, our mother's bodies don't produce those hormones. If there's any perturbation, even a minute, infinitesimally small amount of exposure at a critical period, it can permanently change some developmental trajectory, change molecular processes, change functional outcomes at the end. And there are experimental demonstrations of lack of threshold of endocrine disruptors in a number of different models. We can talk about it later, but I don't believe that there are any thresholds. The other part of that question really speaks to things like dose-response curves. And there's a lot of evidence for endocrine disruptors that they work at exquisitely low doses, and that there are really not predicted monotonic linear dose response curves caused by endocrine disruptors, which is consistent with how hormones act in our body as well. So, great question.